For anybody who's new here, my name is Spencer Fern. I am the science coordinator with Restore the Delta, in charge of this HABS subcommittee, as well as the HABS testing for water quality sampling for our monitoring program in Restore the Delta itself. I guess I'll pass it over to Haley, who's another one of my colleagues in this kind of work, but she focuses more on the air quality, but I'll let her take over from there.
Hi, everyone. Thank you so much for having me. I'm Haley. I'm a PhD student at UNC Chapel Hill, and my dissertation effort has been focused on studying the impacts that harmful blue-green algae can have on air quality. So far we've been working on characterizing how toxins and the cells themselves can get into the air. And my focus is really on, just seeing how much of this is happening in the environment and what weather conditions are influencing that. So yeah, excited to be here and talk a little bit about my work in North Carolina and this study we're starting in the Bay Delta.
Then Stephanie, if you want to pass or introduce yourself.
Sure. Hi, everyone. My name is Stephanie Ng, and I'm an air quality specialist in the Community Strategies and Resources Department at the Valley Air District. And this department just oversees kind of all the implementation of the AB 617 program. And I'll go ahead and pass it to just for transparency, I will say I have two team members from the Air District. One is in the Community Strategies and Resources Department and kind of in training mode right now and observing. And then we have, so that's Eric, and then we have Juan Luis, who's from the Outreach Department. I'll let both of them introduce themselves, but I just wanted to share who's on our team here. And with that, I'll go ahead and pass it over to Eric.
Good afternoon, everybody. My name is Eric Salas. As Stephanie said, I'm the 100% more of her team now, because it was just Stephanie, and now it's me and Stephanie, or Stephanie and I. It's good to meet you all. I look forward to seeing you all in further presentations. Thank you.
And I'll go next. Hello, everyone. This is Juan Luis Carranza. I am the Senior Communications Rep here at the Valley Air District, and I am part of the Outreach and Communications Department, pretty much supporting the implementation of the AB 617 project and working in collaboration with Stephanie and Eric as well. So thank you.
And maybe we'll pass over to Scott from our state agency.
Sure. Scott Wall. I am with the California Air Resources Board, and I work on the Community Air Protection Program, which is the AB 617 program, and I am assigned as the staff liaison to the community of Stockton. And our other kind of lead for this committee is not here today, Eric. I think he's out of the office, and I actually have to motor at 1:30, just so that y'all, if I just mysteriously disappear. So if anything happens between 1:30 or 2 that requires any action from us, please just let me know, and I will hand it over to Douglas.
Hello, everyone. My name is Douglas Vigil. I am a resident and community advocate of the city of Stockton, and I am been advocating for environmental issue, for people with disability. They have having problem with asthma and issue with boundary with environmental and other things. So yeah, definitely I love being part of this meeting and other meetings as well. Thank you.
And then Cynthia or Tanisha, do you want to introduce yourselves, if at all?
Sure, I'll go next. Good afternoon, everybody. My name is Tanisha, and I'm the Environmental Justice Program Specialist with Catholic Charities, and I work along with Jonathan on the AB 617, especially in South Stockton. And I will go ahead and pass it over to Cynthia.
Hi, my name is Cynthia. I work with as a climate water advocate with Spencer with RTD, and I will also be helping Haley with the air monitoring.
All right, so I think that's everybody. Correct me if I'm wrong, but I'm gonna quickly go through what I've seen from HABs testing for the past 2 months. It's been a minute, but just in terms of the fact that Scott, I think you'd want to be here for Haley's presentation. So I'll quickly say that most of the testing we've done the past, I want to say a month and a half, has really only shown that there's been a bloom over in the port of Stockton, going from McLeod Lake to Mormon Slough to the Morelli boat launch. And those are the 3 areas we test, and that's in at least 3 of the 7 areas we test in Stockton have had more bloom activity than the rest recently. As the weeks have gone by, it seems like the bloom is diminishing, and I haven't been seeing too much except for like scattered bits as opposed to the spilled paint I saw initially about a month and a half ago. But interestingly enough, actually yesterday, Monday, we grabbed some water samples from McLeod Lake, even though that the bloom wasn't really visually present, even though, I mean, it was there, it wasn't as visually present. The value for the toxins that we got was above 10 parts per billion, even though the water we grabbed is pretty clear. And that's the first time I've seen that using these types of tests. And just something interesting to look out for. But yeah, it's looking like the bloom is on its way out and it's dying off, which is my theory. And it's what's causing the toxins to be more present in the water. But that's my theory for why that is. And if anybody has any questions, that's kind of a very short summary of what we've seen the past month, but everywhere else has been pretty all right. I mean, the water temperatures have been rising though. It's been 80s in the 80 degrees Fahrenheit, or 26 if we're going to talk Celsius, which I usually prefer, but you know. But if there's any questions, that's pretty much what I have to report. Take that as a no then. Okay. Well, I guess I'll hand it over to Haley then, so she can present what she's been seeing in her air monitoring, or present forward. And do you have a presentation privileges?
I think so.
You should be able to share your
let me
screen.
check here. I only have one screen right now. I'm usually working with 2, so might have some road bumps here, but okay. Can you all see a PowerPoint?
Yes. Yeah.
Perfect. Okay. I will go ahead and get started then. So this is my understanding that this is a subcommittee focused on HABs specifically and how they might pertain to air quality. Is that right? Okay.
Community wise too. Yeah.
Okay. Community wise. Awesome. So I guess, you know, since it's a small group, we can make this as informal as everyone would like. I'm happy to stop maybe halfway through to answer some questions if anything's unclear, but I'll just get through this as quickly as possible and then leave time for discussion and questions if that works for you all. My name is Haley. I am a PhD student at UNC Chapel Hill, and my background is in marine science and biology, but I've recently converted to having a bigger focus on public health impacts of water quality and air quality. So for the past 4 years, I've been researching HABs, specifically these freshwater cyanobacteria and how they might be influencing air quality. So the first thing I want to talk about is just generally how we know that blue-green algal blooms are currently influencing air quality. So the mechanism that causes these cells and toxins to get into the air from the water surface is via some sort of physical disturbance. So this could be wave action that's from wind, or once you get out further into the estuary, this can be tidal wave action. We're not exactly sure how much recreational activity like jet skiing or boating might also be causing some of these toxins to get into the air. This is something I'm really interested in long-term, looking at how maybe fountains or anything that's generating these bubbles that burst, how this could be impacting aerosolization of pollutants. So these waves lead to an entrapment of air bubbles beneath the water surface. And when those bubbles rise to the surface and pop, that's what causes this aerosol, which is a microscopic particle of water that can carry these pollutants and toxins and cells. This is the aerosolization. And, so we're really focused on characterizing what conditions cause more or less of these toxins and cells to get into the air. So this is a phenomenon we refer to as spray aerosolization or primary aerosolization, because the particle itself is being emitted and that particle is carrying the cells or toxins. And so with the blue-green algal bloom specifically, there's been 3 different toxins that have been found in aerosol. So microcystins and nodularin. These are 2 different liver toxins that have been quantified in respirable aerosol. And recently, anatoxin A, which is a neurotoxin, has also been identified in aerosol. So the premise here is that when these toxins are dissolved in the water, the ones that are more hydrophobic or like the air more are more likely to get into the air when compared to their more hydrophilic or water-loving chemical compounds. So the more hydrophobic a cyanotoxin is, the more likely it might get into the air. So these toxins have been identified in these aerosols, and several different aquatic blue-green algae species have also been identified in aerosols. So we can see the toxin translated to the air as a dissolved component of the water, or we can see whole cells of the cyanobacteria getting into the air as these primary spray aerosols. And something...
looks like someone has a question, sorry.
Okay, sure, yep.
Douglas, if you wanted to ask.
Thanks, Minister. Yeah, I mean that the presentation had really kind of shocked me a little bit, but let me ask you the anatine, anatoxin. Are they? You said it was happens in the nerve, neurology. Is that what you mean when somebody goes into the water, or fish, or anything aquatic, it affects the nerves, or what? Just a clarification.
Yeah. Let me just make sure I'm understanding your question. So, are you asking about this neurotoxin?
All the toxin that you just mentioned, and the water that it's out in the air, like boating and swimming and other stuff. That is very bad for people, as well as things that are Toronto or Delta. Has that been happening already, or what?
So in the airshed of the Bay Delta, none of these toxins have been quantified to my knowledge. This has all been conducted in other locations. So we hope to have some preliminary data on microcystin and also anatoxin by the end of this year. So that's what we're sampling for. But I guess my expertise is not what the potential health impacts might be. So even if we do find it this fall, once we get our samples quantified, we're still not sure what concentration is going to be dangerous for human health. And that's not necessarily where our research is focused. So right now we're kind of at the stage within the research of seeing if the problem is even there in the first place. So unfortunately, I can't answer too many questions about, like if we see X amount versus a much higher concentration, what that means from a health perspective. But I guess my area of research is more just looking at, what environmental conditions are promoting their release into the air. Did that answer your question?
Yeah, but how will this interpretation will influence our means? Well, I mean, you have the fact there in front of us, and yet how are we using that as a data for that is toxic water? That's what I'm trying to understand. What the data are they going to be used for if it's not going to be used for a health purpose? Since we in the beginning when we had this presentation, not this presentation, but we had the 6117, our concern was is our health, our asthma is affecting our lungs. It's the way you breathe, or when the smell comes and it's polluted, you should be away. So how that would not bring anything concern on that. That's what I'm trying to say.
Okay, yeah, I think as far as showing data, this information actually comes from studies that I guess aren't mine. So I'm referencing other studies, and I do have like a list of these papers that I will provide at the end, and I can include in the chat if that's helpful, to maybe look further into these different toxins that we're talking about.
Great, thank you so much. Really appreciate it.
Yeah, no problem.
And I'm wondering, Haley, if I could provide a little context, and Spencer and Haley, feel free to correct me. And Douglas, like this kind of field that Haley is working in is definitely like really, really new. And so we know that there are issues with harmful algal blooms in the water causing health impacts. And then now Haley's trying to bridge this gap of understanding if it does happen that any of these toxins in harmful algal blooms can end up in the air. And then that, because this is also new, we don't really understand, one, if it's happening, but then if we do understand if it's happening, what are those health impacts of these harmful algal bloom toxins ending up in our air quality? So this is all, it's really exciting because this is all like a very like new, very like cutting edge kind of research and this is stuff that we're just now exploring. So that's why, unfortunately, at this point, Haley can't really say about, how these toxins can impact that are coming from harmful algal blooms can impact our health from the air quality perspective. We're just not there yet.
Right.
Definitely, thank you. Actually, I couldn't understand better what you just said. Thank you.
Thanks for the great questions and for the chance to clarify. I'll pass it back to you, Haley.
Yeah, so definitely lots of room, questions to be answered for sure in this specific field. So I guess on that note, we've been looking for these toxins because we know when it's in drinking water and recreational water, it can cause human health effects. But there's, tons of different metabolites and chemical compounds that these cyanobacteria produce that we haven't necessarily been looking for yet in the air. And we think that these could also be potential allergens when inhaled. So, we'll be looking specifically for these toxins, but just kind of opening Pandora's box in a way. There's several different ways that these algae might be impacting air quality that has yet to be researched. So in addition to different compounds being emitted primarily just via these physical wave and bubble bursting mechanisms, there's also a ton of volatile gases that are emitted by algae that could potentially form aerosol once they get into the atmosphere. So this is something that we're not necessarily quantifying, but we're working on in separate studies. So just bearing that in mind, these are some of the different ways that we are hypothesizing that these harmful algal blooms are impacting air quality in a way that hasn't necessarily been recognized yet. Okay, so getting into actually showing some concrete findings, I conducted a similar study to what we're doing in the Bay Delta in Eastern North Carolina in the summer of 2020. And some of our major takeaways were that we did not quantify any microcystins in PM 2.5 or aerosol with 2.5 microns in diameter or smaller. So we say this and take this with a grain of salt because we actually quantified very, very little microcystin in our water samples throughout our 4-month study period. But this was good news that we didn't see enough of it to reach our limits of detection in our air samples. But we did also test for cyanobacterial DNA in those PM 2.5 samples, and we found several toxic and bloom-forming type of cyanobacteria in PM 2.5. So we know that their entire cells were getting into the air. We found that when there was more aquatic cyanobacteria blooming in the water, this did influence the abundance of airborne cyanobacteria that we saw. So more cyanobacteria in the water did lead to more cyanobacteria we detected in the air. And interestingly, these 3 species of cyanobacteria that are well known to be bloom forming and harmful were aerosolized at the same time. So when we found higher abundances of Dolichospermum, we also saw higher abundances of Microcystis and Aphanizomenon, which are, as I said, bloom formers. So there was something either occurring in the water or from a weather phenomenon that was causing these to become airborne at the same time. and we couldn't necessarily tease that apart with our data. And then lastly, we saw that the cyanobacterial bloom period was linked to elevated concentrations or mass concentrations of PM 2.5 in the air. And we don't think that this was entirely explained by this primary aerosolization, because when there was more dolichospermum in the water, we didn't necessarily see more dolichospermum in the air, interestingly enough. So we think that there might have been other compounds other than toxins or volatile compounds coming off of these blooms that was also contributing to these increased PM 2.5 concentrations. So this is just one diagram from that study where you can see we binned all of our average median, or I think our median PM 2.5 daily concentrations, and we categorize them by bloom days or non-bloom days. And we found that on bloom days, we did see that increase in PM 2.5. So that's shown in this box plot in panel A. And then in panel B, you can just see, a time series that's showing our PM 2.5 readings. That's the line in correlation with the abundance of dolichospermum, which was the key blue-green algae that was blooming in this system throughout our study, which lasted 4 months between June and October of 2020. So those were the major findings from our research in North Carolina, but discussing what we'll be looking at in the Bay Delta, we are going to be looking at a couple different size fractions of aerosol. So we'll be looking at PM10 and PM 2.5, and we'll be analyzing both of those size fractions of aerosol for microcystin and anatoxin and cylindrospermopsin, which are 3 types of these cyanobacterial toxins. And we're doing this at Discovery Bay in Stockton. This summer, we started in mid-June and we're running through the end of September. And then next year, we hope to start much earlier in the season, hopefully end of April, May, and run through the end of September as well. So we'll be quantifying those toxins in aerosol, but we'll also be collecting a handful of samples to look at DNA and toxin producing capabilities of the actual cyanobacterial cells that become aerosolized. So we're going to be using a metagenomic approach to do this. And then lastly, we'll be collecting water samples and weather data and all sorts of other kind of supplemental data to hopefully, again, try to figure out what environmental conditions are promoting the biggest release of these toxins and cells into the air. So that will be our goal at these sites. So here's just an image of what our equipment looks like. So these giant aerosol samplers have these mushroom heads where the air comes in, and then it goes through a series of tunnels, essentially, that filters it out to be the size that we want, so that we're only collecting aerosol within our size fractions of interest. And then this all gets collected onto a filter, which then we can take back to the lab, extract, and then analyze for those toxins. So these are our PM10 and PM 2.5 samplers deployed at Discovery Bay. And then we have a media intern and science communication intern who's been working with us named Madison Scholes. And she's made these really nice diagrams to hopefully kind of show what's happening in this equipment a little bit easier. So as I had mentioned, we're hoping to get early results sometime this fall. We just received our first shipment of samples and we're going to start processing them in a couple weeks. Just throwing it in that we have made an attempt to create some public facing materials to hopefully spread the word about what we're researching with this project. And our intern Madison has made this really nice graphic to try to show what we're trying to accomplish with these giant foreign looking, like alien looking type of samplers. So we have this pamphlet that we're hoping to print out, and then this QR code also will lead people to a website where they can learn more information about harmful algal blooms. There's a link to the state's HABs reporting flyer, and then there's just information about air quality and how HABs can impact air quality. And then this actually has nothing to do with our data, our research, but I did want to bring up just other pertinent information. A study last year was published looking at the efficiency of different household filters for your air conditioner, and also different face masks and how they do in terms of filtering out these cyanobacterial toxins from the air. And this group at Miami found that high-rated air filters and also KN95 and N95 masks are effective at blocking out microcystin carrying aerosols. So if you live near the water, if you're going to be near the water for whatever reason, you can use this PPE to protect yourself from these toxins. Okay, so I think I talked much more than I anticipated, of course, but that's all I have. I just want to acknowledge all of the awesome collaborators we have on this project. It's a really big group of a big collaborative team working together to make this possible. So our funding source is the Bay Delta Council. We have co-investigators from UNC Chapel Hill, North Carolina State, UC Santa Cruz, and the University of Miami. And then we're also collaborating with a handful of the governmental organizations studying water quality in the region. So USGS, California Department of Water Resources, state water boards. Of course, Restore the Delta, Spencer, Barbara, and Cynthia have been amazing at loaning us some of their time and resources. And then I have an amazing team within my lab group who's been spending time with us out in California and helping me back here in North Carolina get things up and running. So with that, I'm happy to go back through anything and answer questions. And again, I'm hoping to share this reference list, and I can do that as soon as my presentation ends. And then if anyone is interested in checking out our public-facing website, you can scan that QR code, or I can send you a link. So that is all.
Looks like we do have a few questions.
Okay, let's see,
yeah. I think that I saw Douglas raising his hand earlier in the meeting.
I am so happy with your presentation. I really think that it's wonderful to learn from you. I learned a lot. So that's good. The thing I'm more concerned about, when you showed the slide, the presentation, and you showed the machine, the mushroom, the only thing in the background I was more concerned about those boats that might cause some effect, like there's some cooking, the propane, and the other smoking marijuana, whatever. If that's going to affect the machine or whatever, can you will it be any different if you have that machine away from the people, away, so you have more air quality that kind of stir multiple of a mixture of kind of where the air goes from north, south, east, and west? It that would be more helpful than just leaving it right by the dock or something like that. That's what makes me concerned. That's all.
Yeah, that's a great point, in that our machine is essentially like a giant vacuum. So it just sucks all the air across a filter. But so we will collect any other type of particles that are in the air. And this could be smoke particles, or it could be exhaust particles. We will be collecting all that. But the method that we have in the lab to test for just toxins will ignore all of that stuff.
Oh.
Yeah. So that allows us to distinguish all that junk and the toxins we're interested in.
Okay, then you do have a feature where you kind of ignore other element and focus on what you want to look at, concentrate on the factor. Oh, okay. All right, that's cool. Okay, thank you.
Yeah, so looking at the chemical makeup of it allows us to do that distinguishing. Yeah.
Thank you.
Yeah, no problem.
And then Tanisha, did you have a question?
I did, thank you. So one question that I did have was, Haley mentioned that the two sites were from Port of Stockton. So my question was, what makes these two sites prone to algal bloom relative to the other area?
Yeah, great question. So I think the biggest issue here is the stagnation of the water in these respective sites. So, I'm not too familiar with the geography of the Delta as a whole, but I know Discovery Bay especially has really, really slow water movement through it because, you know, all the hydrology has been modified to kind of make this canal system. And I'm sure everyone who's from this area, correct me if I'm wrong, like Port of Stockton is the same way, and that like there's not a lot of water coming in and flushing through here. And this is really what these toxic cyanobacteria love, is a really hot, stagnant water body. So that's allowing for these spaces to be hot spots.
Okay, thank you. So I wanted to know if you could email me this information. I'll drop my email in the chat.
I think I just caught the tail end of that, but I heard email in the chat, so we can definitely.
Yes. Okay, thank you.
Yeah.
And Haley, if you feel comfortable, any part of that presentation, any like outreach materials, I know you had that QR code and the really cool graphic and the story map, any of that you're able to share, we can post it on our website too, to share out with our greater CSC.
Okay. Yeah, that would be awesome. Yeah, I can share the presentation and then these different materials as well. I'll follow up with you.
Cool. And I wondered if I can also add on to kind of the point that Douglas made about, I know that you all had faced kind of challenges in terms of like just like what it's like to monitor in like North Carolina versus like Stockton. I'm wondering if you want to add a little bit to that, just in terms of, because it sounded like in terms of the filters and how much pollution was getting caught and how long they could run. What's the unique challenge here?
Yeah, so interestingly, like I've been checking in on the purple air maps because that's kind of my quick and easy way to figure out like how much particulate matter is floating around. And I think like this time of year, it's actually we've had worse air quality in North Carolina. So I think what we had done 2 years ago in North Carolina prepared us fairly well for this site. So like the hours of the day that we're running our samplers, so far it hasn't led to any issues in terms of like over clogging our filters or anything like that. So far, hopefully, you know, if there so happened to be like a fire that we were, you know, getting a lot of the biomass and smoke coming from, that we might have to pivot the hours of the day and shorten the time periods that we're collecting. But as of right now, everything is going great.
I just had a general question. I know Discovery Bay's got both the PM10 and the PM2.5. I know Stockton's only got PM10. Is there a reason behind that, or was it?
So the only reason is that we could only afford one for the other site before the funding officially started on July 1st. So we have that other PM2.5 sampler sitting waiting for us for next summer.
Gotcha.
We were finally able to order it. So that was just out of lack of resources than it was.
PM10 was the more
need. Yeah.
Gotcha.
Yeah.
Okay.
I don't want to take up too much space, but maybe I could just ask in terms of this like research that's going on. So you've just kind of finished up this research that happened in North Carolina, and now are working on this in the Bay. So it sounds like it'll span a couple of summers of data collection before you are kind of ready to publish. Is that right?
Yeah. So I think, you know, this summer has been a great pilot summer, and like, you know, getting our feet wet, figuring out what's going to work for us, like conducting a lot of this research remotely. And of course, like you all are making it possible, like having some like boots on the ground. But yeah, so we hope next fall to like start actually doing the hardcore analysis. You know, we'll get our preliminary data now, see if we need to pivot any approach for collection next summer. But as far as like early results, I don't think that will come until the end of 2023.
May I ask why that long, the end of 2023, we're in the middle of 22? How long did it take? Are you referring to the data that you're receiving in the end of 23? Or are you going to place the monitoring filter at the end of 2023? I'm a little confused between that. So when are you going to have that there? Really, and we can make a note of it. Thanks.
Yeah, so we go out and we collect data for like 4 to 5 months at a time. So incrementally, we're getting tiny bits of data, but we really don't like extract and process all of the samples until the very end of the season, just for like resource purposes. It's much easier just to kind of do it all at one go. So that's the reason it takes so long, is that we won't have all of the data collected until the end of next fall in 2023, which is why we will start processing samples and getting those early findings then. But yeah, as I had mentioned, we're hoping to get some early data on this summer by this fall, because similar situation, we'll take everything that we collect between June and September of this year and actually run it on our machines in the lab in October timeline. So that's about when we hope to start sharing, like whether or not yes or no, we found some sort of toxins in the air.
Any more questions?
Thank you all so much for your time.
Thanks.
I really appreciate it.
Thanks for being willing to present. I know it's almost 5 o'clock for you out there.
That's okay. Yeah. It's a good end to my day.
Right. All right. Well, if that is it, not entirely sure when the next HAB subcommittee should be, whether it should be the end of the month or maybe the end of this month. But generally speaking, I tend to try to keep it towards the end of the month because it's easier to present the data I've gotten the past month. But beginning of the month works. It's kind of the same concept. I'll just be going over what I did the month prior. But other than that, I appreciate everybody coming out. Thanks, Haley, for being willing to present, and hoping this helped answer some of the questions about how we're putting our air monitors as well as the water quality all together so that it's something we can present to the community. So thanks for the time.
Thanks all for joining. And yeah, Haley, please feel free to send us any of the materials. I can drop my email into the chat too.
Okay, perfect. Yeah, I think I have it. So I will follow up with you.
Cool.
All right.
Thanks,
Great.
everyone.
So nice to meet all of you.
Guys, everybody take care.
Bye, everybody.
Bye. Thanks.